Hello, good afternoon, everyone. We are going to be doing lecture 2 this week, and the topic is blood and lymphatic system. So let's get right into it. Over here, you can see here's a picture of the blood, and you have the red blood cells here, and there's a white blood cells here, and you can see, they do look quite different, not only in the color but the shape as well. So as we go through the lecture, we are gonna learn more about the characteristics of each type of cells, and we will also learn about their functions. So blood. There are approximately 5 liters of blood in an average 70-kilogram individuals. Of course, that does vary from person to person. That's just like an average number, right? And blood essentially can be described as a protein-rich fluid with cellular components. Okay, so the fluid is mostly water. And in that water, we have a bunch of proteins that are dissolved in it. As you will see, there are also minerals and nutrients and some waste product that are dissolved in it as well. And then if you throw in some blood cells, then essentially you have blood. Now, we can take blood and separate it into its individual component. We can do that by spinning it at very high speed. I'll show you a picture of that in the next slide. And once you separate it, you will have the protein-rich portion at the top because it's lighter. Okay, and that is what we call the plasma. Okay? Plasma makes up approximately 55% of your total blood volume. The cellular components, which we call the formed elements, those are heavier and they sink to the bottom of the test tube, and that makes up approximately 45% of your total blood volume. So you take blood from a person, you will put it in a test tube. We actually, this picture is not entirely accurate. When you take blood from a person, it usually goes directly into the tube. Okay, it's one of those vacutainers, right? You will know when someone take blood from you, right? They have these little containers with like purple caps or yellow caps, or sometimes green caps, right? And then they would take the blood directly into those containers, and then you can spin it down at really, really high speed. And as I said, you will get the fluid portion with the proteins in it. That's the plasma on the top. And at the bottom, you will have your formed elements. So the formed elements are all the cells found in the blood, including red blood cells, RBC is red blood cells, white blood cells, as well as platelets. Okay, if you are not familiar with these cells, cell types, we're going to be talking about it. Okay, they all have like a fancy name other than the regular red blood cell name. For example, erythrocytes, that's red blood cell. Leukocyte, that's white blood cells. And platelets, we're just going to call it platelets at this point, okay? But they do have another name as well. And don't worry about it at this point. When we talk about the individual cells, the names will come up again. But I'm just trying to tell you, right? There is this common name, if you would, and then there is like the biology name, if you would, for the cells. So red cell is a majority of your blood cells. Okay, they make up the bulk of your formed elements, and you don't have a lot of white cells or platelets, and they only make up less than 1% of your whole blood. And in fact, it's lighter. Okay, the white cells and the platelets are lighter because it doesn't have the iron in it, and so it's hovering between the plasma and the red cell portion. There is a question: should you memorize all the names? Yeah, I mean, like you should recognize them. Like if the question talks about erythrocytes, then you should know it's referring to red cells. Okay, I know it's a lot. All right, so here we have 2 pictures. Well, we have 3 pictures. On the left side, this is what it looks like when you spin down the blood. At the top here, that's where the plasma is. And at the bottom, that's where the formed elements are. Formed elements. Okay, and hovering here, which is also part of the form element, that's where you will find WBC and platelets, right? Now we're going to learn about blood transfusion later on today. And I want you to understand that when we do blood transfusion, we have to separate the plasma from the red cells. Okay? Plasma and red cells, the transfusion rules for them are actually different. You cannot just take a person's blood and then transfuse it directly to another person. That just not going to work. Okay, even if they have matching blood type, is not necessarily going to work out. Okay, so like in movies, you see person hooked up a line directly to another person. That's not really how blood transfusion work. Okay, and hopefully after this lesson, you will understand why that is not the case. Okay, like you hear people say, oh, I'm O negative, and then they can give it to everybody, right? But again, that's not necessarily true. And I'm gonna try to explain all these things to you throughout the lecture. So let's take a closer look at the plasma. Okay, the plasma is like the liquid portion, right? And I forgot to mention actually that these plasma and then the red cells, once you separate them, you can actually store them. Okay, plasma can be stored for months. Okay, you can actually freeze the plasma. It's no problem. But then the red cells, you can store them in refrigerators about 6 degrees for up to 42 days. And then they are no longer good for use. Okay, you can also separate the platelets. The platelets can be stored at room temperature in an agitator. So like you have to keep on moving them for up to 5 days. Okay, so platelets are good for clotting your blood, right? So there are actually a lot of different blood products that you can create from the whole blood, right? And a lot of times you don't necessarily have to give whole blood to the person. It depends on what the person needs. Then you have to combine these things and transfuse to the person. So in the plasma, like I said, is mostly water, and there are some dissolved ions in it. So some important ions in our blood include things like sodium, chlorine, chloride, potassium, magnesium. All these things are going to be found in the plasma portion of the blood. There are, of course, useful nutrients as well. So nutrients like sugar, right? There are some amino acid, right? And there are also waste products. Okay? Waste products like CO2, maybe some urea from protein metabolism. And that's why the blood needs to be cleansed, right? That's why the blood needs to be filtered at what organs filter our blood? Does anybody know that?
Lungs?
Not quite. Well, okay, I see what you mean. Like the lungs will help get rid of the carbon dioxide, yeah. But what other organs will help specifically get rid of the unwanted material from the blood?
Liver?
Yep,
Kidney?
that's right. The liver and the kidney, they both are capable of removing quite a bit of waste product from the blood. And then there are a bunch of plasma proteins. Okay, you already know what proteins are. You learned that last semester from me, right? Proteins can be hormones, right? Communication between different parts of the body. You have proteins that helps you fight infections. You have proteins that help you stop bleeding. All these proteins are dissolved in the plasma of the blood. Okay, so we call them plasma proteins. Okay, plasma proteins. The other part of the blood is the formed elements. Formed elements. Right? That's like the bottom portion of the centrifuged blood sample. After you spin it down, the bottom portion is the formed elements. So we have the red blood cell, white blood cells, and platelets. Now this is where things get a little bit confusing. Okay? Because there's a lot of red cells in the formed elements, we actually give it a special name. This whole part, like all the red cells collectively, it's called the hematocrit. Okay, hematocrit. And that's all the red cells. The individual red cells are the erythrocytes. Okay, but like when you pack it together in the test tube, when you spin it all down like that, then it's called the hematocrit. The main function of red cell is to deliver oxygen, and actually they help deliver the carbon dioxide back to the lungs as well. But we'd say deliver oxygen. The white cells, the white blood cells, there are a lot of them, and we will learn each one of them in a bit. But collectively, the white cells, they help defend the body against pathogens. Pathogens are things like bacteria, virus, fungus, fungi, right? Parasites. Okay? All these things are basically like living organisms that can cause you to become sick. So your white cells are there to help defend the body against these pathogens. Okay. And the last part of the formed elements would be the platelets. Okay. The platelets are there to help stop the bleeding. If you have a cut, the platelet will seal the cut and form the scab and stop the bleeding. Okay, so that's just like a really brief overview of what the red, white, and platelets do in the body.
Vincent, does the plasma do anything?
Yes. The plasma has the nutrients and the ions, right? So as the blood circulates through the body, the plasma basically helps distribute these important ions and these important nutrients, right? And as you will see, because the plasma contains these proteins, all these proteins are going to play a role in different areas. Okay, some proteins are going to help you with your immunity. Some proteins are going to help you with blood clot. Some proteins are going to help you communicate between different organs. Okay, so it's not so much what the plasma does, but it's the stuff that's in the plasma that will do all these things, if that makes sense. Okay, so let's take a closer look at the plasma proteins. We'll be learning, quite a bit, quite a number of plasma proteins as we move through the course. And here I'm just going to give you like an overview of what they actually are. Right? So the most abundant plasma protein, the one that you have the most in the blood, okay, is something called albumin. Okay, albumin. So albumin is made in the liver. Your liver is on the, like right here, right, top right a quadrant of your abdomen, and it makes a lot of plasma protein. And one of the plasma protein that it makes is this thing called the albumin. And what albumin does is, as far as we know, it stays in the blood and it basically adds to the concentration of the blood. By adding to the concentration of the blood, you are able to maintain the blood osmotic pressure and therefore the blood volume. Okay, remember, we learned this last class, right? Your capillaries, your blood vessels, they have these solutes in there, right? There's the dissolved ions, there are the cells, but more importantly, we have these albumins in them. Okay, so like albumin in them. Right? That would help further increase the osmotic pressure. That way you will be able to draw fluid into the blood vessels and keep it there, right? To maintain the blood pressure and the blood volume. Okay. So like I said, we make our own albumin, But people who take protein shakes, the way that they eat, it's basically albumin. Okay, you will find them in baby formula and maybe in egg whites, right? There's these albumin proteins in there. Now, if you look
Professor,
at, yeah,
sorry, excuse me.
no problem.
The albumin, is that the albumin make osmotic pressure in the blood?
Yeah, so albumin is not the only thing that creates the osmotic
Yes.
pressure. Everything that dissolves in the blood helps contribute to the osmotic pressure, right? But
Mm.
albumin is a major contributor, because you have a lot of it, right?
Okay. Yeah.
Yeah. So if you think about kids who are malnutrition, right? You might see these pictures in World Vision posters or something like that, right? One of the characteristics of these kids is that they have a big belly, okay, big tummy. They are skinny everywhere except for the tummy. And the reason for that is because when they are malnutritioned, that means they don't have enough proteins in the diet, right? For example. So without adequate protein in the diet, they don't have the amino acid to make the albumin. Right? So when they don't make enough albumin from the liver, their osmotic pressure actually will go down because now you have less things in the blood, right? So then you're not able to retain as much fluid in the blood vessels. And again, last lesson, we learned about capillary exchange. While osmotic pressure is trying to pull the fluid and hold it in the blood vessels, what force is pushing the blood out all the time? What force
Blood
opposes?
pressure?
That's right. The blood pressure is pushing it out, right? So here, if these kids don't have enough albumin in their blood, then the blood pressure is winning, right, so to speak. So they end up losing a lot more fluid than they are able to bring back in, and all those excess fluid is now accumulating in the abdomen. Okay, and that's why you see this like swelling in the belly, right? Now you might wonder, how come it doesn't, cause other parts of the body to swell up, right? Like why not the legs? Why not the arms? Well, we also talk about how when you move your arms and move your legs, the muscle actually help return some of those fluid back into circulation, right? But there's not a lot of movement in the abdomen, right? Like, we don't really contract the abdominal muscle regularly, right? Like when we just walk around or sit down. And that's why those fluid will stay in the belly and not go anywhere. Okay, so that is albumin. It's one of the most abundant plasma proteins. Okay, what other plasma proteins do you have? Now, this is just an overview. We're gonna go into more details later on, okay? So don't feel overwhelmed here. Just get an idea of what types of proteins there are. There's another question. Stomach bloating is the same thing? Not quite, okay? Stomach bloating is like when you have a lot of gas generated from certain kinds of food, right? Some people, when they eat pepper, they have these bloating in the stomach because of gas buildup. This is fluid that's inside, okay? It's the leaking of the fluid from the blood vessels because of the reduced osmotic pressure. Okay, so other plasma proteins include ones that are necessary for blood clotting. Okay, so I already told you, like platelets play a role in blood clots, but it doesn't do it alone. Okay? There needs to be some proteins to help you along the way. Okay, and here are two examples. One of them is called fibrinogen, and then the other one is called prothrombin. Okay, I know it's a lot of terms, but just recognize it for now. I'll explain more later when we get there. Antibodies, you probably have heard of antibodies a lot, like when you receive a vaccine, the vaccine is going to help your body make antibodies so that it will help you defend against say COVID or like the flu, right? So antibodies are these proteins that you make. In fact, they are Y-shaped proteins. And that's going to be a theme that we're going to be talking about. Y-shaped proteins that help you defend the body, essentially. Please do not confuse antibodies with antibiotics. Okay, I found that people get those two confused sometimes. Antibiotics are medicine that you take to fight off bacteria. Okay, antibodies are different. Like your body makes the antibodies to help defend it. Okay, so let's say you have chicken pox, then your body will make antibodies to fight off the chicken pox. Then those antibodies will stay in your body and you'll be protected for the rest of your life. Okay. So again, the purpose of this slide is just to show you that there are different types of plasma protein. Some will play a role in helping you stop bleeding. Some will help you fight off things that are not supposed to be in your body. Any questions so far? Okay. Now, so we're gonna switch back to the blood cells now. And where do blood cells come from? Okay, the red cells, the white cells, the platelets, where do they all come from? Well, the process of making new blood cells is called hematopoiesis. And hematopoiesis is creating, you can say forming new blood cells, not just the red, all of them. Okay. Hematopoiesis. All right. So that happens in bone marrow. Bone marrow occurs in bone marrow. As we age, we don't have as many active bone marrow sites anymore. Okay, so in adults, right, like our femur, like your thigh bone, the bone marrow in there, that's gonna make some new red cells for us. Okay, like our sternum's gonna make some new red cells. Like over here, you have a bone called the ilium, right? The bone marrow in there will also help you make new blood cells. Okay, but that's about it. There's a limited sites for red cell for blood cells creation in adult. Okay, so you don't have to memorize these things, but it shows you in the bone marrow you have a multipotent stem cells. We learned that, right? Last lecture of last semester, right? That these stem cells are capable of becoming various types of cells. Okay? So depending on what kind of stimulation they receive, they might become a red cell, they might become a platelet, and then they might become, all these different kinds of white blood cells. Okay? So creating new blood cells, we say hematopoiesis. Okay, hemato is blood, poiesis is to create. So creation of new blood cells. So now we're gonna take a closer look at the cellular component. This is actually my blood right here. I think last year I was having like a really severe allergic reaction of some kind. And my wife, her job is to look at people's blood. Okay, so she took some blood and wanted to check it out for me, and it turns out like the cells that fights off like when you have like allergies, those kind of cells was really elevated in me, and that was causing like all the rash and stuff like that. So anyway, she took a picture of my blood and gave it to me because she thought I would be interested in showing my class. So you can see these are the white cells, and the purple ones are actually, sorry, the red ones are the red cells, RBCs, and then the purple ones are the actual white blood cells. Okay, they're not actually purple. Okay, like they are purple because you can color them, right? You can stain them when you prepare these kind of slides. And do you guys see the platelet here by any chance? Anybody want to guess which one might the platelets be?
The ones with hollow center?
Oh, okay, yeah, actually the ones with the hollow centers are also red blood cells. Okay, it just depending on like what angle you're looking at them at. Sometimes, you know, if they're tilted, then the light pass through them easier. That's why they appear to be hollow. Yeah, the little dots, that's right. Okay, so like that's the platelet. Right? So from this picture, you can see red cells are really common, right? White cells are not as common, and the platelets are also not very common. Yeah, this picture is real. Right? Someone messaged me as, "Is this picture real?" Yeah, this is real. This is my blood. Okay, a blood sample from me. Okay. So let's take a closer look at red blood cell, okay, or RBC. Okay? The biology name, okay, for red blood cell is erythrocytes. Cytes means cells. Okay, erythro means red in Latin. Okay, a lot of these words are based on the Latin language. Okay, so erythrocytes are red blood cells. They do not have nucleus in them, and without the nucleus, it allows the cell to kind of pinch in at the center. Okay, so that creates what we call like a biconcave shape. If you look at the red cells from the side, you know, it's pinched in at the center. Okay, so that's called concave. That's concave. If it's concave on both sides, then it's biconcave. By having a biconcave shape, it creates more surface area for gas exchange. Okay. Inside the red cells, there is a protein called hemoglobin. Hemoglobin has 4 polypeptide in it, right? It has a quaternary structure. Again, that's something we learned last semester. And that is the oxygen carrying molecule in RBC. People who have low levels of hemoglobin or red cells, or if they have defective hemoglobin, then they would have anemia. Okay, so if you don't have enough hemoglobin, or if you are making the wrong kind of hemoglobin, like if there's something wrong with it, some kind of defect, because you have a mutation, that could lead to anemia. Okay? But it's not just the hemoglobin that could create an anemic situation. People who don't make enough red cells, that could also cause anemia. Now, there are different causes for anemia. Do you remember one example, one type of anemia that we learned last semester? I mean, I must have repeated several times.
Sickle cell.
Sickle cell anemia, that's right. So sickle cell anemia is a point mutation that result in a missense mutation. So in the original amino acid sequence, we have a glutamic acid here in position number 6, but after the mutation, it becomes valine. You don't have to memorize this, okay? And that is enough to cause the hemoglobin to misfold, and ultimately it causes your red cells to change its shape into sickle cell shape. Right? These cells are not very good at transporting oxygen. They get stuck in the capillaries, and the person becomes anemic. So shortness of breath, they get tired easily. And these sickle cells, when they get stuck sometimes in the capillaries, they could create quite a bit of pain as well. Okay, so this is a recessive mutation. So for people who have normal red cells, they are like big S, or big S, small s. To get sickle cell anemia, it would have to be little s, little s. Both copies, both alleles would have to be mutated on the chromosomes in order for the person to have sickle cell anemia. Okay? So it turns out if a person has sickle cell anemia, they are immune to malaria. Can someone tell me what malaria is? Like what spreads malaria?
The fever
Mosquito.
and
Mosquito?
Yeah, mosquitoes, right? Okay, and it could cause a person to have these high-grade fevers, and then they could potentially die from it. Okay, so malaria is very common in these regions as shown on the map. And oops, sorry, wrong map, on the left map here. Okay, so it shows in Southern Africa it's very common, the malaria, and in some parts of Asia as well. But it turns out if you have sickle cell anemia, you are protected from malaria. And so what's really interesting is if you look at the genetic makeup of people who in populations who are found in places with a lot of malaria, majority of the population is big S, small s. Okay, so if you're big S, small s, you have a little bit of the sickle cell trait, but not enough to make you sick from sickle cell anemia. So you're not going to be like super anemic. Okay, you're pretty much normal. However, because one copy is the sickle cell shape, it also gives you protection from malaria. Okay, so like you get benefits from both of the phenotype. Okay, so
is that the AS genotype? The AS.
yeah, the big S, small s. Yeah, that's right. Right. So again, the big S gives you normal red cell. The small s, it causes some cells to be sickle cell shape, which protects you from the malaria. But because you do have some normal red cells, it's not going to give you actual anemia. Does that make
Okay.
sense? Yeah, so it's
Yeah.
like best of both worlds, right? Okay. Now, that's not the only cause of anemia. There are other anemia out there, like hemorrhagic anemia. That's like if a person is bleeding too much, right? There is anemia caused by, say, chemotherapy, because chemotherapy suppresses your bone marrow and you don't make enough red cells. That could cause anemia as well. And then there are, anemia related to, not being able to absorb, say, vitamin B12, right? So, like, I mean, you don't have to know all of these. My point is, there are different causes of anemia, but ultimately, it's because your hemoglobin is being affected or your red blood cells is being affected. Okay, that's basically what it is. Okay,
I
so
have a question,
yeah,
Professor.
sure, please.
So, most of people, or I don't know, here in my country,
Mhm.
they related anemia with lung.
With
So,
what? I'm sorry.
it's lung, LUNG.
LU. Oh, anemia with the lungs, related to the
Yes,
lungs.
yes.
Um,
Is that any related or not?
I am not familiar with a kind of anemia that's specifically related to the lungs. But I can look into it. Okay.
Okay.
But generally, people who have anemia would have some kind of respiratory symptoms, because when you are anemic, you are not like getting enough oxygen, right, to your system. So like, there's going to be shortness of breath, like I said, right? You might feel like laborious in your breathing, right? So that's the only connection I can make at this point. But maybe
Okay.
there's more to it. Okay. Thank you.
Okay.
So the red cells, where do they come from? Earlier I told you they come from the red bone marrow stem cells. Okay, you have these stem cells that will undergo the hematopoiesis, right? Let me write that down for you again, so you will recognize the word. Right? So that's the process to make more blood cells. And when your blood cell level declines, when there's not enough red cells, Okay, maybe you just donated blood, maybe you have have some bleeding, and now your RBC levels are down. Then your kidney will actually release a hormone called EPO. Okay, EPO stands for erythropoietin, but you can just call it EPO. Okay? And the EPO will stimulate the bone marrow to make more red cells. Okay, so one more time. When your red cell level drops, the kidney makes EPO. Then EPO goes to the bone marrow, and it will help you make more red cells. Okay, bring it back to normal. Okay? So athletes, sometimes athletes, they go up to high altitude to train. Right? In high altitude, the air is thinner, and there's not as much oxygen, right? So your body will feel the strain. You're not able to deliver as much oxygen to your cells. And so similarly, the kidney will release the EPO, and then the EPO will make more red cells, and the athlete gets adapted to the higher altitude, right? And then when they come back to sea level, for example, they have all these extra red cells in the body, which will make it easier for them to compete now, right? It's like better than before, because now you can deliver oxygen even more efficiently, right? So you can actually, like not you, but then like pharmaceutical company, they can make EPO in a bottle, and then they could sell it, and it will be used for cancer patient. I told you, cancer patient undergoing chemotherapy or radiation therapy, they are typically a little bit anemic, okay, because their bone marrow is being suppressed. So you give them a shot of EPO to boost red cell production. EPO can also be used for people undergoing dialysis. Okay, dialysis is for people with kidney problems, right? Kidney failure, for example, they're waiting for a kidney transplant. So in the dialysis treatment, they have to take the blood out of their body and then basically clean it and put it back into their body, right? But sending the blood cells through all these tubings and all these, filters and stuff like that can destroy a substantial amount of them. And so the person might become a little bit anemic, right? So you give them a bit of EPO as well. Okay, that's that.
Vincent, so how do they give them this EPO? Is it via the veins or?
Yeah, injection, exactly. Just injection.
Okay.
Mhm,
Okay.
yeah.
Okay.
So,
Vincent, I have a question.
yes, yep.
First, can you back? Okay, so can we say whoever gets dialysis because their kidney is not working, is not functioning to produce this EPO, they get this EPO
Oh,
from
I'm so
outside
sorry.
as well?
I didn't clarify enough. Okay, so dialysis is not because the kidney doesn't make EPO. Okay, dialysis is when the person's kidney is not working in the sense that it's not filtering the blood to make the urine. Okay, so
I got the concept that you said, but I'm saying because the kidney is responsible to produce EPO, right?
yes,
So
yes,
when somebody is gets dialysis, it means the kidney is malfunctioning, right?
that's
And
right.
then the EPO is not produced as well, right? Am
Okay,
I
so yeah,
right?
so the kidney for dialysis, okay, for dialysis, the kidney is malfunctioning not because it's not making EPO. It's malfunctioning because it's not filtering the blood to make the urine. Okay, so because it's not cleaning the blood, that's why in dialysis you have to take the blood out of the body, clean it, and put it back, right? So this is called dialysis. But when you take the blood out and clean the blood, in the process, you actually killed off a lot of red cells. So then the person might become a little bit anemic, okay? And so to compensate for the anemia, you have to give them EPO as well. Is that a little bit better?
So yeah.
Okay,
Okay.
so again, right, we are giving them the EPO not because their kidney is not making EPO. Okay, we're giving them EPO.
Okay, if somebody's kidney is like destroyed, okay, so in like the kidney cannot produce EPO as well, right? When the body needs it.
Okay, it may be okay, but again, that's not the key issue here. The key
Okay,
issue is the
I got
kidney
it.
is okay. All right, sorry if I'm not answering your question. Just send me an email, okay? I'm happy to clarify it for you. Okay, so how long do red cells last in the body? It turns out not too long, okay, about 120 days. So that's like, roughly 4 months. And when the red cells are old, they will be captured at the liver and the spleen, where they will be eaten up by something called macrophage. Okay, so macrophage is a type of white blood cells that we will look at later. But they're basically there to eat up these worn out red cells. Okay, so liver is top right quadrant of your body. I showed you already, right? Like right here, that's where the liver is, and then the spleen is right here. Okay, like at the back here, top left quadrant, behind the stomach. Okay. When you break down the red blood cells, the hemoglobin is released. Right? So hemoglobin is made up of proteins, and it also has iron in it. Okay, iron. And so you would recover the protein, you recover the iron, and return them to the bone marrow to use it for making more hemoglobin. And then whatever is not recycled, it will get broken down into something called bilirubin. Bilirubin. Okay? So bilirubin is yellowish, yellow in color. And we normally would get rid of the bilirubin through urine and feces. And that's why the urine and the feces are like the color that they are. Okay, it's because of the bilirubin. Now, sometimes kids are born like with a yellow skin tone, right? Does anybody know what that condition is called?
Jaundice?
That's right, jaundice. All right, so jaundice is actually a buildup of bilirubin in the skin. Okay, and we're gonna talk about that probably not today, but like next class. Right? But remember, bilirubin is a waste product, is a byproduct of hemoglobin breakdown. Okay, that's it for red cell. Any questions before I move on to the white blood cells?
Yes, Professor. Bilirubin is you said the waste
Yeah,
product.
it's a waste product. It's a byproduct of hemoglobin breakdown.
Okay.
Yeah. Okay. Hold on a second. I'm just looking at this. So yeah, by the way, next Tuesday is going to be your quiz number 1. Okay. I'll post the information later today. Okay, but we will still have a class on Tuesday. Okay. So you make your timetable. Okay, set it up so that you can do the quiz on your own. And Miran has sent me the rest of the schedule already. So I will post this on eCentennial after class today. Okay, so in case you want to know what's going on for the rest of the class of the semester, it's going to be available to you. All right. Moving on, white blood cells. Okay? White blood cells is also called leukocytes. Leuko is white, okay? White cells. They are also produced in the bone marrow, just like the red cells, just like the platelets. All blood cells are produced in the bone marrow, right? They came from the same stem cell. Stem cell would become different things depending on what the stimulation is. And the process, once again, is called hematopoiesis. Okay. Now, white cells are a little bit different from red cells. Red cells are only found in the blood vessels. But white cells, they are found in the blood vessels, but they can also squeeze themselves out of capillaries and stay in tissue fluid or in some organs. Right? For example, they can stay in the liver and the spleen to help destroy these old red cells. Okay? So they are in the bloodstream, and then remember the capillaries are leaky, right? So the white cells can squeeze themselves out, and they could like attack, things that are not supposed to be there. Their job.
Vincent, when you say blood vessel, you mean the atria, ventricle, and capillaries, right? Those three.
Okay, so when you say atria and ventricles, those are chambers, okay? Chambers of the heart.
Oh,
When
okay.
we say blood vessels, we're talking about like arteries, capillaries, and veins, right?
Okay.
Yeah,
Thank
yeah.
you.
But the white cells will only be able to squeeze themselves out at the capillaries. The capillaries are leaky, right? Not the arteries or the veins. Arteries and veins, they're seal tight. It's only the capillaries that are leaky. So the white cells, they play a role in tissue maintenance and repair, as in just making sure there's nothing that's not supposed to be there in your tissue. When I say tissue, I mean like your body tissue, right? Like tissue in the kidneys, tissue in the liver or something like that, right? So you just want to make sure that they are free of pathogens. Okay, if there is any kind of damage, they would initiate some kind of repairing mechanism. They, as in the white cells, will also help destroy dead or dying cells, like the red cells that we talked about. But other cells that are too old, they can be taken up as well. They could be eaten up by the white cells. So as I said, red blood cells are confined to the blood. They are going to stay inside the blood vessels, but the white cells can squeeze themselves through the pores in the capillary walls. Okay, and once they come out, they can stay in the tissue fluid, or they could stay in an organ. Okay. All right, enough talking. Let's.
Sorry, I have a question.
Sure.
Do the white blood cells, after they do their tasks out of the capillaries, do they go back to the capillary or
Yeah,
not?
so usually they don't, okay? Usually they come out and then they fight until they die, okay? And the ones that stay in the organs, they will basically eat up as much as they can, and then they will die off there. All right, so they have a limited lifespan, right? Okay, so they don't usually return. Yeah. All right, enough talking. Let's write some notes. Okay, just switch it up a little bit. Less boring that way. This is class notes May 14th. All right, so here we go. White blood cells. Okay, so if you have your paper, we're gonna write white blood cells here, and then we're gonna separate into different categories, and under each category, we're gonna have different things, okay? Just so that you can plan your space accordingly. So white blood cell. Right? And the fancy name for white blood cell, just take a moment and try to recall it in your head. It is the leukocytes. Leuko is white. Okay. Erythrocytes, that's the red one. Leukocytes, the white one. Now the white blood cells, we can classify them based on what they look like under the microscope. Okay, now these cells are like transparent for the most part. Okay, like if you look at it under the microscope, like there's like almost no color to it. They're so thin, right? Right? Like the picture I show you, the purple and the pink, that's because we stain them. Okay? The stain we use is called H&E stain. Okay, that's like the most common stain for when looking at tissue under the microscope. Okay, so some cells would take up the pink really well, some cells would take up the purple really well. Okay, so then we can classify the white cells into two categories. Okay? We have what's called like granulocytes, and the opposite of granulocytes is the same word, granulocytes, but with an A in the front. Okay? So granulocytes and agranulocytes. Okay? What's the difference? Granulocytes are cells, white cells, with visible granules under the microscope. Okay. When you look at them under the microscope, you can see they have these very distinct granules. Okay, so like here's a cell, and then you will see the nucleus here. A lot of these white cells have like multiple nucleus. Okay, nuclei, and then you will see these like granules. Okay, these little granules right there. Okay, it's very obvious when you look at it under the microscope. On the other hand, the agranulocytes, they are cells with no visible granules under the microscope. Even though they have them. Okay, so both have granules. Granules contains these, proteins and chemicals and stuff like that. It's almost like a lysosome, if you would. So they have it, but you just can't see them. Okay, for some reason they don't take up the dye as well as the granulocytes. So this classification is based on how they look. It's based on appearance, okay, rather than say functions. Okay, granulocytes, you can see the granules clearly, and the agranulocytes, you can't. All right. Now, let's take a look at some examples of granulocytes. Okay, number one, it's called neutrophils. Neutrophils. Okay, neutrophils, they make up 70% of your white blood cells. Okay, now remember, you don't have a lot of white cells, right? Anybody remember the percentage that I showed you earlier today? Like if you spin down the blood, how much of it is actually white cells?
All of them are 45%, yes?
Oh, okay. 45% is white, red, and platelets all together.
Yes.
Yeah. But what about the white cells alone? What percent?
Less than 1%.
That's right. Less than 1%, right? So in that 1%, 70% of it is going to be the neutrophils. Okay. So neutrophils, they are phagocytes. You remember phagocytosis last semester? Right? They go around and eat things, right? That's what the neutrophils are. Right? They move around the body and then they eat up anything that is not supposed to be there. Okay, they see a bacteria, no problem, let's eat it up. They see a dying cell, let's eat it up. Okay, so that's neutrophils, makes up 70% of all your white cells. Okay. My friend's son, when he was younger, he didn't have enough neutrophils. Right? They weren't sure why. Right? And so he gets sick all the time. He was not allowed to go to daycare. His immune system was really weak. Yeah? But as he grew up, went back to normal. So neutrophils, important.
So, Vincent, how the white blood cell recognize that is like virus or bacteria, or
Yeah,
that is
very good question.
white cells?
Yeah, excellent question. We actually will learn that next week when we learn the immune system. But long story short, we actually talked about this last semester as well when we say membrane proteins, right? One of the functions of membrane protein is cell-cell recognition. Okay, so your white cells can identify a cell based on what's on the outside. Okay, if a cell has something that's on the surface that's different from what you have, then it knows it's not part of the body, and then it's going to attack it, right?
Yeah, thank you.
Yeah. Okay, so second one. Now, some things are too big. You just can't eat them. Like the cell is too small and the pathogen is too big, like a parasite or something. Okay, it's not possible to eat them up, and so you need a different kind of cell to deal with it, and that is called eosinophil. That's how you say them, okay? I swear. Eosinophils. Okay, so eosinophils, they will fight parasites. Okay, now I am keeping this very simple for you, as in like I'm just giving you like a one single function. Okay, do yourself a favor, don't read this online, okay? Because they actually do a lot more. You're gonna get overwhelmed, right? But like if you do read across something other than fighting parasites, it's probably true, okay? But for the purpose of our course, I'm just telling you that it fights parasites, okay? So like if you have a parasitic worm or something in the body, like that's like really big. You can see it with your naked eye, but the cells are too small. You cannot eat it. So the eosinophil will fight the parasite by secreting chemicals. Okay. Number 3, we have another one. It's called basophil. Basophils. Okay. So basophils, they are responsible for triggering inflammation. Triggering inflammation. So you know inflammation, right? Like when you cut your finger, it's gonna be painful, it's red, it's hot, okay, and it's a little bit swollen. All these are characteristics of inflammation, and that is brought on by the basophil. Okay, so eosinophil and basophil, they actually cause a bit of allergic reaction as well. Okay, so like when I had the rash last year, and my wife looked at my blood, it was that I have more than normal eosinophil and basophil. Okay, and so by analyzing a person's blood, seeing what's high and what's low, you can understand what kind of problem the person is having. Okay, so these are granulocytes. Okay, you can see their granules under the microscope. All right, next we have agranulocytes now. Okay, agranulocytes, these are things that you cannot see the granules, even though they have them. So the first one is called a monocyte. A monocyte. A monocyte is, you can think of them as like inactive. Okay, they're just like, moving around the body, making sure everything is okay. And when they encounter something that shouldn't be there, they can transform into macrophage. Okay, so macrophage is the active one. Okay, so this transformation, we have a name for it. It's called differentiate. Okay, so the monocyte can differentiate into macrophage. It's like a one-way transformation. There's no turning back. Okay, so they turn into the macrophage, and they will fight the infection. Sometimes for organs that are particularly vulnerable, like the lungs, okay? Because we keep on breathing things in, right? So there might be pathogens in the air that we breathe in. So it's a very vulnerable organ. Then we would put some macrophage there just in case. Okay? We call them resident macrophage. They basically live there. Right, because they know it's inevitable that you're gonna be breathing in some pathogen. Right, so just gonna stay there and clear them as they come in. Okay, and then you have lymphocytes. Lymphocytes. Okay, there are two kinds of lymphocytes. Okay? There are B cells, or B lymphocyte. It means the same thing, and T cells. Okay? You might have heard of these. During the pandemic, maybe. B cells, they make antibodies. All right, so we talked about antibodies a little bit earlier today, right? They are these Y-shaped proteins that will attack whatever pathogens you have. Okay? If you have chickenpox virus, then you're gonna make antibody against the chickenpox. If you have the flu, you're gonna make antibodies that will fight off the flu virus. And then we have the T cells, which will help coordinate immune response. Okay, think of them as like the general in a war. Right? Like, okay, you go attack here, you go defend here, you do this, you do that. That's what the T cell is gonna do. Right? It's gonna help direct other immune cells. So there you have it. Okay, I know it's a lot, but keep this chart handy with you. So let's take a look at what they look like under the microscope. Right, so that's a neutrophil, that's an eosinophil, that's a basophil. Okay, and people can train to recognize them, right? And then you can count them, see what's high, what's low. You have to know what's within a reasonable range. Okay. All right. So formed elements include the red cells, white cells, and now we can talk about the platelets. The fancy word for platelets is thrombocytes. Okay? Thrombocytes. I actually don't know what thrombo means, okay? But some kind of cells, okay? Platelets. So the platelets are different in the sense that they are not really cells themselves. Right? Rather, they are fragments that is broken off from a much bigger cell called the megakaryocyte, okay? Which is this guy right here in the middle. Okay? Mega is big, right? So this megakaryocyte, bits and pieces break off from it, and that's how you get the platelets, okay? Platelets all over the place. You make a lot of platelets every day, okay? 200 billion platelets per day. They don't have a nucleus, and they are only good for 10 days. Right? After 10 days, they are broken down, reabsorbed by the body. And we need the platelets for blood clot. Okay? Fancy word for blood clotting is hemostasis. Hemostasis, not to be confused with homeostasis, right? Okay, let me write the other word. Okay, don't confuse with homeostasis. Okay? Homeostasis is maintaining an internal balance, right? So that could be body temperature, calcium level, blood calcium, blood sugar level, so on and so forth, right? This is hemostasis. Make sure you don't bleed out. It's very similar, the word. All right. We're going to take another note quickly, and then we'll have a break. Okay, coagulation.
Then I have a question. So platelets are kind of cells, not particle or
Oh, they are
molecules,
cell fragments. Yeah, cell fragments.
cell fragments.
Like bits and pieces of a bigger cell. Yeah.
Okay.
Yeah.
Vincent, so this also forms part of the 45%, right?
Yes, yes.
Yeah.
Yep,
Okay.
yep.
So
But
so
it's all but it's like less than 1% in total, right?
okay.
Yeah. Let me show you quickly, okay? Just to, I know there's a lot of numbers. Okay, so like here, this thin layer here, that's the white cells and the platelets. It's less than 1%.
Okay.
Yeah.
So if we use a microscope to check this on out, we are going to find these big mega karyocytes. Is that?
The megakaryocyte is actually super rare. Because
Okay.
the megakaryocyte is gonna be broken down into the platelets, right? Okay, so like you're not very likely to find a megakaryocyte. It will be there, but then it's like a rare find. Okay.
Okay.
Because most of it would have been broken down into the platelets, right? Does that make sense?
Yes.
Now, in fact, you can see the red cells here. Like these are the red cells. Okay. Now they are not red anymore because they use a different dye, right? You know what I mean? When you make these slides, you have to use the right stain. Yeah. Anyways, coagulation. All right. So let's say this is a blood vessel of some kind, and the blood is passing through it, no problem. I told you blood is okay. They are happy if they are passing through a smooth surface, right? Smooth surface. And the inside of the blood vessel is smooth because it's made up of the endothelium, right? All this is in the lecture last week, okay? So let's say you have a thumbtack or something, some kind of cut, okay, some kind of thing that damages the blood vessels. Right? So now it exposes the underlying tissue. Okay, so damage to blood vessel exposes underlying tissue. So these damaged tissues that are exposed, they release a chemical. Okay? Release something called thromboplastin. Okay? Thromboplastin. Okay, so these thromboplastin are usually like nicely hidden away, but it's going to be exposed when you have a cut blood vessel. Okay. Now, very quickly, just going to go back to the PowerPoint slides here. Earlier, an hour ago, we were talking about different types of plasma protein, and there are two kinds that are needed for blood clotting, right? Fibrinogen and prothrombin. Okay, so in your blood right now, in your plasma, you have some prothrombin. You got some prothrombin. If you see the prefix pro, more often than not, it means it is inactive. Okay, so the prothrombin is inactive, and you want it to be inactive normally. Right? If you don't have any cuts, you don't want your blood to clot, right? It's bad to have a blood clot in the middle of your blood vessel for no reason, right? That could lead to like a stroke or something, right? Actually, that does happen, right? It's called an embolism, right? You can look that up. But normally we don't want that to happen, and then it just stays inactive. However, when you do have a cut, then the cut is going to be releasing the thromboplastin, and then the thromboplastin, along with some calcium, is going to activate the prothrombin, and it turns it into thrombin. Okay, so this is green light now. This is activated. Active. Okay. And also in your blood right now is fibrinogen. Okay. And again, if you see the suffix ogen, it means it's inactive. I'm not making this up, right? Like for real. Okay. If you see pro, if you see ogen, it's inactive. You're gonna learn a few more words in the course that have these, prefix and suffix. Okay, it's inactive. So to turn it on, you need the thrombin with some calcium, and then that will turn it into fibrin. Okay, fibrin. So once you get the fibrin will form these long strands. Okay, that will help basically seal the plug here. Plug the leak, I guess. That's the right way of saying it. It's going to seal the leak, okay? Plug the hole. Right? By forming these nets, then you will be able to trap some red cells, and you'll be able to trap some platelets. Okay? And if you trap enough platelets, you will form the scab, and then you will stop the bleeding. Okay? That's basically like an oversimplification of what happens. So there will be a question on the test that asks you to reorganize the steps. Okay. So again,
Vincent, can you explain just one more time?
Sure, yeah. So it will ask you to reorganize the steps. So first of all, when you damage the blood vessel, the underlying stuff is exposed. Okay, so it released this thing called thromboplastin. Okay. Thromboplastin will activate prothrombin, and then it turns it into thrombin. And thrombin will activate fibrinogen, which turns it into fibrin. And then the fibrin will form these nets to trap the red cells and the platelets. Okay. So again, in your bloodstream right now, in my bloodstream right now, assuming we don't have any cuts, like at the moment, if we don't have any cuts, then the prothrombin and the fibrinogen would just be circulating around our body. It's in your plasma, okay? So when do we need them? We only need them if we have a damage, right? Okay, when we have some kind of cut. And to trigger this thing to start happening is to have the thromboplastin. Okay, so once you have the thromboplastin, it kind of switch on one thing, and it's like domino effect. You push over the first domino, the prothrombin becomes the thrombin, which then turns the fibrinogen into fibrin. Okay. There's a question. Prothrombin is inactive. How are you asking me how prothrombin gets converted into thrombin? Is that the question? I'm not quite.
Yes, Professor.
Yeah. So a lot of time these are proteins. Okay, the prothrombin and the fibrinogens, they are proteins, and a lot of times there is like at the end of the protein there is a few amino acid that's blocking its function. So if you cut them off, then it gets turned on. That's usually how it happens. Okay.
Professor, what about
So-
calcium? Tell me what
Oh, what's the
do
function of the calcium? Yeah, the calcium, a lot of these things like requires like enzymes to perform the reaction, right? As you learned last semester, and a lot of these enzymes actually require ions to we call them cofactors. They're like little helpers to help with the reaction. That's probably the simplest way to explain it. Okay.
Thank you.
Yeah.
Vincent, so when the blood vessels are damaged, what triggers the release of this thromboplastin?
Yeah, it's once it's damaged, like all the stuff that was nicely hidden away before is now
Okay.
exposed, right?
Mhm.
So that's the stuff that's releasing the thromboplastin.
Oh,
Okay,
okay.
it's like think of it like this, like there's a lot of stuff underneath the carpet, but as long as the carpet is there, we don't see it, right? But if you peel off the carpet, like all the fluffy stuff, whatever, like that dust and everything comes out, right? That's kind of like the thromboplastin coming out. Do you understand?
Yes, thank you.
Okay. All right. So again, I'm not making this up. This is for real. This is a real picture that shows you the fibrin. That's the white strands here, and it's trapping the red cells and a little bit of platelets, and that's the thing that's going to be sealing the plug, sealing the hole. Yeah, I don't know why I keep on saying sealing the plug. It doesn't make sense.
Vincent, back to the notes. So,
Yes.
so once the thromboplastins triggers these dominoes, this prothrombin, and then becomes active, is the next step fibrinogen, or it's just another domino on its own?
Okay, so release thromboplastin, prothrombin becomes thrombin, and then the fibrinogen becomes fibrin. But in order for the fibrinogen to become the fibrin, you need to have the thrombin.
Oh, okay.
Okay. So, give you a little bit of like a real life example of how these all connect. When a person goes through surgery, they actually have to check your PTT. It's called the prothrombin time. They need to figure out how long it takes for you to stop the bleeding. Okay? If it's outside of normal range, then they have to give you transfusion of some of these clotting factors. Right? And that's an example of where you might need to have like plasma transfusion, okay, before the surgery. Because otherwise, it's dangerous to go into surgery if your blood is not clotting fast enough, right? You understand me? Okay, so this is a summary of everything we talked about for the past hour and a half almost. You don't have to memorize the numbers, but the functions are all here, and these are all the cells that we talked about, okay? Very quickly, a few poll questions, and then we're gonna have a break. We are running a little bit behind, so I am just gonna, you know what? Let's do a pause, but try not to take too long, okay? Okay, everybody who chose something got it right. Example of a granulocyte, that's the basophil. A basophil is for inflammatory response. Next one. Okay, very good. The answer is parasitic infection. It's right in the notes. Last one. Hemoglobins are found in the red blood cells. Very good. Okay, see, not bad, right? Not too bad. All right, let's take a break before we learn about the genetics of blood. 5 minutes, guys, as usual. Okay, see you in 5 minutes. Okay. You know what? I will leave the note on, leave the screen on this thing here in case you want to take a closer look. Yeah, see ya. All right, let's keep going. So now we have a basic understanding of what's in the blood. We are gonna shift gear and talk about the genetics related to the blood. So there's gonna be a little bit of overlapping with the genetic stuff that you learned last semester. With the genotype, phenotype, dominant, recessive kind of stuff. Okay, so here is a picture of a red blood cell. And on the surface of a red blood cell is actually very busy. You have all these different molecules on it, okay? And these molecules, some of them are proteins, some of them are sugar, some of them are sugary proteins, and they collectively contribute to your blood type. Okay, so let's take a closer look at that. Blood type refers to the inherited antigens that are present on the surface of the red cell. So two things here. Inherited means you are born with your blood type. You don't get to change it. You don't get to choose it. You're born with whatever you have. And it's inherited from your parents. And these are antigens that are present on the surface of the cell. Okay, so antigens are basically stuff that is on the surface of a cell. But when it comes to blood cell, we are specifically looking at the antigens that are on red blood cell. Okay. Let me say that one more time. All cells have antigens. Okay, your muscle cells have antigens, your liver cells have antigen, virus have antigens, bacteria have antigens. Okay, when we talk about blood type, we are looking at the antigens that are on the red blood cell specifically. These antigens could be proteins that are on the membrane. It could be carbohydrates. Glycoprotein is like sugar protein, right? Sugar protein. Glycolipids would be like sugar lipids. And collectively, there are over 40 different types of blood group. Yeah, there's a link here. You can click on it. And it will take you to Wikipedia, and there's all the different blood groups here. Okay. So anybody know their blood type here and want to share? I mean, you can make it up. I would never know, but I just want to hear some blood type that you are familiar with.
B plus.
Yeah.
A plus, O.
Yeah, exactly.
B,
Right. So
and B.
What you guys is telling me is the ABO blood group, right? You see on the screen here, it says ABO blood group system, right? And when you say you are plus or minus, you're actually talking about the rhesus blood group system, okay? Which are the two things that we're gonna look at in the lesson, okay? ABO and rhesus. But there are other ones. Look, there is MSN, right? There is like the P1PK, whatever, right? So these are like blood group system, or sometimes they say antigen system. It's the same thing, okay? It's like just talking about the things that are on the surface of a cell. Here's a cow, here another antigen system, and so on and so forth. Okay, so a lot of different things that are on the surface of the red cell. Now, over here, according to the Canadian Blood Surface, we have majority of the population is like O positive, right? 39% of the population, followed by A positive. That's another common one, right? B negative and AB negative are a little bit uncommon in the human populations in Canada anyways. Yeah. Now, does it give you benefit to be a certain type of blood? Nah, not really. Okay. The only thing that we have to worry about is when it comes to transfusion. If you have a really rare blood type, it might be more difficult to find the suitable blood to transfuse for you. Okay. Other than that, there's no health benefit to be like B positive. There are books out there that says, if you have a certain blood type, you should follow a certain kind of diet. I don't think those are good science necessarily. Okay, I don't think there's any science behind those, in fact. Okay, moving on. ABO blood type. Right. So ABO blood typing is based on the presence or absence of two possible antigens. We'll just call them antigen A and antigen B on the surface of red cell. Now, these antigens are based on the kind of alleles that you inherit from your parents. And in the population, there are 3 alleles for the ABO blood group system. Okay. It's IA, IB, and little I. And so I'm going to write you another note to help you understand more. I always mess up this part of the note. Hold on a second. I just need to take a look at the slides ahead so I don't end up writing things too early. All right, here we go. Blood group, blood type, ABO blood type. Okay, 3 alleles in humans for ABO, and the alleles are in the PowerPoint slides, right? IA, IB, and little I. Even though there are 3 alleles that exist in the population, how many different alleles can you have at most in your cells, in your genome?
2.
Yeah, because we are diploid, right? We only have 2 sets of chromosomes. We only have 2 slots. So what are the combinations? Well, you can be IA. That's homozygous IA. You can be IA little I. You can be IB, IB little I. You can be IA, IB, and you can be little I, little I. Okay. So IA and IB are dominant over little I and codominant over each other. That means IA will cover up the little I, but when you have both IA, IB, they will both show up. Okay. So if the genotype is IA or IA little I, then the phenotype is type A blood. So if I draw my red blood cell, I would have the A antigen on the surface. This is red blood cell, okay? With the indentation in the middle. For IB, little I, this would be type B blood. And then the blood cell, the red blood cell, will have the B antigen on it. And what if someone is IAIB? What do you think the blood type is going to be?
AB.
That's right. Because A and B are codominant with each other, right? So on the red cell. I'm sorry, it's like not very clear here. Let me just make them smaller. Okay. So for the type AB blood, there is going to be both A and B on the surface. Little I means you cannot make A, you cannot make B. So if you don't have A and you don't have B, it's called type O. Okay. So no A and no B. That's called type O. Are we okay with the genotype and the phenotype here before we move on? You sure? Okay, so let's do a genetic problem. This is from the PowerPoint slides. Example 1. A man with AB blood is married to a woman with AB blood. What blood types will their children be and in what proportion? Okay, so the man is AB blood, so the genotype has to be IAIB. There are no other possibilities. For the woman, she is also IAIB, right? And I hope you remember the Punnett square from last semester. So if the man is IAIB, half the sperm will carry the IA, the other half will carry the IB. Same thing for the woman. Half the eggs will carry IA, the other half will carry IB. And so if I finish the Punnett square, it will look like this. Okay. Again, this is not new material. I mean, the concept of blood type is new, but then the Punnett square and everything, it's what you learned last semester already. So help me out here. What's the percentage? What's the percentage for the blood type? Anybody?
IAIA is 25%,
25% type A, very good. And
type AB 50,
Mhm.
and type B is 25.
Right, 50% what? I'm sorry, I missed that.
AB.
Yeah. Right, these ones, right? And then 25% type B. Is everybody okay with this setup? You should ask now if you are confused. It's only going to get more difficult. Okay. Let's take a look at the next question. So let's say the babies got mixed up at the hospital. Okay. So can we find out which baby belongs to which parents? We have two parents here, two couples, and then we got two babies. So give you a moment to try to figure it out. Anybody? Which couple does baby one belong to?
Second one.
Second one, yeah. All right, let's see. So you shouldn't be doing the Punnett square here. Okay? If you're doing the Punnett square, the approach is, I mean, you will get the answer probably, but then it's like gonna take too long on the test, right? This is what you're supposed to do. Okay, let's write out the genotype for type O. There's only one possible genotype for type O, right? Little I, little I. If you don't remember what genotype corresponds to what blood type, you can take a look at this note here, right? So type O is little I, little I. What about type A? Well, for type A, it is going to be IA something. I don't know if it's IA or IA little I. Not enough information. Let's move on to the Jameses. Type B is IB something. Again, I don't know if it's IB or IB little I. And type AB is IAIB. And that's where we can stop. And we can see the answer at this point now. Mr. James is type AB. So he's only going to be able to pass on either IA or IB to the kids. Okay? Can only pass on IA or IB to his children. And baby one is type O blood. In order to make a type O baby, each parent needs to contribute a little I. Mr. James does not have a little I to pass on, so he cannot have a type O baby. For that reason, the type O baby belongs to the Marshalls. And then baby number 2 belongs to the Jameses. Okay. Let's think about that logic again. If he's AB, it's not possible for him to have a type O child, regardless of who he marries. Okay, the wife can be O, the wife could be AB, the wife could be whatever. It doesn't matter. Because if you are AB, you do not have a little I to pass on. And you need to pass on a little I to make a type O child. Any questions on that? Okay, so let's write that down. A person with AB blood cannot have a child with type O blood. Again, it doesn't matter who you marry. You do not have a little I to pass on. That's why you can have a type O child. Okay, so someone's asking me, like, then how is it possible for these people to have a type O baby? Okay, well, it's possible if they are IB little I. Right? If you do the Punnett square for IB little I, there is a 25% chance for type O child. Okay, does that answer your question?
Professor, both Mr. James and also have type B can have IB with I.
Say
Small
that
I.
for Mrs. James?
Yeah, when
Yeah,
Mr.
sure. Okay, so let's say she has little I, right? But that's not enough to make a type O baby, right? To make a type O baby, each parent needs to pass on the little I. So even though the mom
I
can pass on, sorry?
I mean, I always come with the B. Is small I come with B?
Oh, it might be, it might not, right? It's like she can be IB or IB small I, right? Right? Homozygous or heterozygous.
Okay.
What's the question again? Maybe I'm not addressing the.
But Professor, both they have type B, but how the child come with I, small I?
For who? For the marshals?
Yes.
Okay, so if we go back to here, there are two ways to have type B blood, right? You can be IB, or you can be IB small I, right?
Okay, you should always refer to this chart.
That's right. Yeah.
Okay.
It's
Okay,
only
thank
when you are AB, then you guarantee IB. When you are type O, then you guarantee little I, right?
Okay, thank you.
Yeah.
Got it.
Any other questions? Can I have another one of these statements, please? A person with AB blood cannot have a child with type O blood. Can I have another similar statement? What kind of blood type cannot have what kind of blood type? Help me out here.
A person with type A blood can with IA cannot have type B blood.
Okay, but like if she marries someone who's like AB, then it's possible, right? You know.
Okay.
Yeah. Anyone else? Okay. What about a person with O blood? What kind of kids can they not have? Regardless of who they marry.
AB blood.
That's right. Cannot have a person with AB blood. Right? Because to have a child with AB blood, you have to pass on IA or IB, right? But if you are type O, then all you can pass on is little I, right? If you're type O, you can only pass on little I. It's not possible to make a type AB baby, regardless of who you marry. Do you guys understand this? I feel like I'm not really
No, this type O is a bit confusing.
yeah, type O
Because
is small I, right?
small I, yes. So
So
the AB blood is type capital IAIB, right?
that's right. So
Okay. So if I marry someone, let's say I'm type O and I marry someone with IAIB.
sure, yeah. So let's do the Punnett square and see, right?
Okay.
Let's do the Punnett square here. Do you agree that's how we set up the Punnett square?
Yes.
Okay, so and then these goes here. So look, 50% will be type A, 50% will be type B.
Yeah, right.
Right? So not possible to have an AB baby because I can only pass on a little I, right? The other parent cannot simultaneously pass on A and B to the kid. They can only pass on A or B, and then the other parent needs to supply the other half, right? But if the person is type O, again, they can only pass on the small I, right? That's gonna take some time to fully grasp, which is okay. We have time. Let's do another example. Okay. So here's the Shrek family. It's an old movie. I don't know if you guys all know it. But this is a pedigree, right? Pedigree. We learned about pedigree last semester. Family tree. Square is male, circle is female. Same generation have kids together. Okay, so I want to figure out the blood type of everybody, the genotype of everybody in this family. Okay, I'm going to give you a moment to do that. Okay, so that was like 3 minutes. Does anybody have the blood type for Fiona that they want to share?
IA small I.
IA
Have-
small I. Okay, Mary, did you have the same thing or?
Yeah.
Yeah, okay, so let's see, right? So the way to approach a question like this is to start with the obvious blood type, like the O and the AB, right? So here Fargo is type O, so he must be little I, little I. For the king, it's IAIB, and that's about it. Right? Those are the only individuals that we can be certain about their blood type. For everyone else, we can write out half of it. Right? The queen is IA, but again, I don't know if she's homozygous IAIA or heterozygous IA little I, right? So I'm just gonna write half of it for everyone else. Okay. Then let's take a look at Fargo again. He has a little I. One of them must came from the father. In this case, Shrek. The other one must have came from the mother, Fiona. Right? Remember, you inherit one from each parent. Now we can continue to trace this back for another generation. Where did Fiona got the little I from? Well, not the king. The king only has IAIB. So it must have came from the queen. Now let's take a look at Fergus here. Fergus has an IA. Now, where did the IA come from? Well, it's not Shrek for sure, because Shrek doesn't even have an A in his genome, right? So it must have came from Fiona. And in fact, we can say for certain that Fiona got the A from the king. Okay? Even though the mom also has an IA, but you know, she didn't pass it on. She passed on the small I instead. Now, what's the other half of the Fergus genetic makeup? Well, he already got the IA from Fiona. That means from Shrek, he's gonna get either the IB or small I. And in this case, it has to be the small I. It wouldn't have been the B, because if it was the B, then the blood type would have been AB, right? And not A. Similarly, for Felicia, I am running out of color, but the IB has to come from Shrek, and then the other one would be from Fiona. Okay, she wouldn't have got the IA from Fiona, because again, that would change her blood type, right? And that's how you do a question like this. Any questions? Okay, let's do an other one here for practice. All right, maybe we'll talk about this one. So same story. We start with people that are either type O or type AB. Those are the easiest to work with because there's only one possible genotype for them. Okay. So John and Harriet, they are the parents of all these people. So Sue is little I, little I. That means one little I must have came from Harriet, and then the other must have came from John. Let's take a look at Mary here. Mary is AB. The A came from Harriet because Harriet couldn't have passed on the B, right? She's type A. And therefore, the B must have came from John. So John is IB little I for sure. The next question asks about Mona. Okay, next question asks about Mona. It's the same family tree. So I'll give you a minute or so. Let me know what you think about Mona.
Vincent, you didn't put up the polling.
Oh yeah, I wasn't going to because then there was like only 2 people was participating, but okay, I'm gonna relaunch it. Thank you. Okay, well, more people chose something this time. Very good. So Sue is little I like before. We already know John is B and I, right? Okay, and Harriet is IA little I. So Ed is little I. And what about Mona? Well, Mona for sure is gonna have IA as part of her genotype, but the other one, we don't know. We don't know what it is. Okay, a lot of people chose IAIB for sure. You got to read this properly. John is not their son. John is the son-in-law, right? They are not related by blood. Okay, only Harriet is their daughter, right? Otherwise, like they're like the siblings are marrying here, right? Like that doesn't make sense, right? So we don't know what Mona is. Mona is IA something. That other something could be IAIB, IAIA, IA small I. We don't know. So the answer is need more information. Okay. Is that why like a lot of people chose A here? Is it because you guys misread that John as the son? That's probably why, right? I'm guessing.
Yes.
Yeah. So just be careful on the test. Okay, moving on. Antigens and antibodies. There is a table on the study guide, second page, number 6 here. So I'm gonna copy the table over, and then we're gonna fill it in. But before that, I want to clarify, reiterate, whatever you want to call it, some terms again. Okay, so antigens. Antigens. Okay, so these are molecules. Excuse me, molecules found on cells. In the context of blood groups, we are looking at antigens on RBC specifically. Okay, so antigens are just molecules on the cell. They are not good, they are not bad, they are just there. Okay, all cells have it. Your cell, my cell, bacterial cell. When we talk about blood type, blood groups, we are talking about specifically the antigens that are on RBC. And then there is antibodies. Antibodies are Y-shaped proteins found in which portion of the blood do we find the antibodies? Does anybody remember from almost 2 hours ago now?
B cell,
Yeah, okay, the B cell makes them. And sorry, the other person, what did you say? Where do we find them?
Lymphocytes.
Okay, yeah, the lymphocytes, the B lymphocytes makes them, but where in the blood do we find it? Which portion of the
And
granulocyte,
granulocytes.
okay. The Y-shaped proteins are found in the plasma, guys. Right? Plasma. Let's go back here just to show you so that you remember where to find it later. Right here. Okay. Some plasma proteins are important in immune response while others play a role in coagulation. Antibodies found in the plasma. Okay. But you guys are correct. Like the B cells, the lymphocytes, they make the antibodies. But once you make them, they're going to be in the plasma. Okay. Y-shaped proteins found in plasma, and your body, or you can say the body makes antibodies to target foreign antigens. So I just pause for a second and listen. Okay, this is very important so that you don't get confused. I keep on saying your cells have antigens, my cells have antigens, bacterial cells have antigens. We all understand that, hopefully, right? Antigens are just molecules on the surface. But the kind of antigens I have on my cells are not the same as the kind of antigens that you have on your cells. Okay, no two people share the same antigens on the surface of the cells unless they are identical twins. That's why you can't just take a kidney from anyone and then transplant it into someone else. Right? The person will reject the organ, right? Because it would treat it as foreign. It would treat it as something that's outside of the body. So anytime you have something that is outside of the body, your body will make antibodies to attack them. That's what this is saying. The body makes antibodies to target foreign antigens. Questions on that before we dive deeper into it? Okay, so let's fill in this chart. We already have part of the answers, okay? but it doesn't hurt to do it again, I suppose. So if someone is type A, they are either IA. Remember, like these are alleles, right? These are genes. Genes makes protein. So basically, if you have the IA allele, it helps you make the A protein so that you can put it on your red cells. Okay. So IA and little I. The little I doesn't make anything. Okay, but that's okay. If you have one copy of the A gene, you can still make A protein. It's not about more or less, right? It's not like you make more A or less A. It's like if you have A, then you have A. So the antigens on red cells, right, is just going to be A. Try to use color. It will make it easier for you to follow. Now, here's the million-dollar question. What kind of antibodies will a type A person make? What kind of antibodies will a type A person make? Remember, you make antibodies for things that is foreign, for things that is not inside your own body. So in this case, you will make antibody that attacks B. Okay, this is called anti-B antibodies. So people with type A blood, they will have anti-B antibodies in their plasma. Okay? Next one. We'll do this later. Next one. IB or IB small I. So it's going to be B on the surface. And help me out here. What kind of antibodies will a type B person make?
A.
Yeah, anti-A, right? Anti-A, exactly. Okay. You always make antibodies for things that you don't have. Okay. Genotype for type O is little I, little I. Little I is not is a gene, but it doesn't make anything. Okay. So on the surface is nothing. No A, no B. There might be other stuff from other antigens group or other blood group, but there's no A or B on it. So what kind of antibodies does a type O person have in their plasma?
A and B.
That's right. Yeah, so they have two kinds. They have anti-A and anti-B. Because they don't have A and they don't have B. So they're gonna need to attack both A and B. Okay, the Y is the antibodies, okay? They are actually Y-shaped. AB type is IAIB. These people have both A and B on them. So what kind of antibodies do they produce?
Nothing.
Nothing. That's right. Nothing. No antibodies. Okay. Otherwise, if they make anti-A, they will end up attacking their own cells. If they make anti-B, they will also end up attack their own cells. Right? Now, I want to clarify here. Okay? When I say no antibodies, I'm talking about antibodies for A and B. Okay? Like obviously, if this AB person gets, again, my favorite example, if they get chicken pox, they will make anti-chicken pox antibodies to fight off the chicken pox, right? Okay? So when I say no antibodies, I'm just talking about like the ABO blood group. Is this clear for everyone? Let me know if you have questions. It's only going to get more confusing when I talk about blood transfusion now. Blood transfusion is the last thing we're going to learn, and then we're gonna call it a day. Any thing you want me to clarify? Okay. Now, at the beginning, I told you we don't just transfuse whole blood. Whole blood means you take the blood out without separating them. That's called whole blood. You cannot give whole blood to another person directly. Okay? You have to separate the blood, right? Separate into different blood products, right? Separate into red cells, the hematocrit, separate into the plasma. The donation rules, the compatibility rules for red cells are different than the rules for plasma. In the past, I teach both, and people get like super confused. So for your sake and for my sake, we are just going to stick with RBC blood transfusion, okay? We are only talking about the rules for transfusing the red cells, okay? But in reality, people will need to have red cells as well as plasma in some cases, okay? But we are just going to be doing the blood, red blood cells. So on the test, if you see a question talking about transfusion, you can safely assume that we are talking about red cell transfusion. Clear? So we have a donor. The donor is the person who's giving the blood, and the person who's receiving the blood is called a recipient. Recipient. I think I spelled that one. Recipient, P-I-E-N-T. Yeah. So the donor is donating the blood, giving the blood, and the recipient is receiving the blood. There are 3 questions we need to ask ourselves. Okay, I'm gonna try to go through the explanation with you first, and then I'm gonna give you like a shortcut, okay? For those of you who don't want to think too much. So number 1, we want to know the antigen. The antigens on donor. Donor is RBC. Number 2, we need to know the antibodies in recipient plasma. And number 3, we want to know will the antibodies attack the RBC. Those are the 3 questions we need to think about when we determine whether the transfusion is safe or not safe. So let's say we take type A blood. Let's say we take type AB blood, and then we give it to type A. Okay. I want you to think about it. Do you think this is safe or not safe? You don't have to tell me. You can just think about it. Okay. Safe or not safe? Okay. So you write it down on paper, and then I'm gonna go through these questions and then see if you got it right. So first, this is the donor. This is the recipient. First question, antigens on the donor's RBC. So because the donor is AB, you can look at the chart. Their RBC is going to have A and B on it. Okay. We don't have to worry about the donor's antibodies because those are not being transfused, right? Remember, the antibodies are in the plasma. After you get the blood from the donor, you got to separate it into red cells and the plasma. We're not talking about the plasma. Right? The antibodies are in the plasma. We don't have to worry about it. We are only giving the red cells. So that's that. Number 2, the antibodies in the recipient's plasma. What kind of antibodies do we have in the recipient? Recipient is type A, so they have anti-B antibodies. What do anti-B antibodies do? Anti-B antibodies will attack anything with a B on it. So in this case, they're going to attack, and it's going to destroy the cell. This is not safe. Do you understand that? Please ask questions if this is confusing.
Can you repeat, Professor?
True. Let's try another example, okay? To see. Let's do O to AB. Okay? O to AB. Let's do a poll here. Do you think this is safe or not safe? If you think it's safe, please choose A. If you think it's not safe, please choose B. If you're still not sure, please choose C. Okay, well, let's take it up. So, antigen on the donor's RBC, this is type O, so there is no A, there is no B on it, which is fine. Antibodies in the recipient, this person is AB, so there's no antibodies. Will the antibody attack? No, because there are no antibodies. So this is safe. This is safe. Let's just do one more. What about A to AB? Can you do A to AB? Well, A has A on it. AB has no antibodies. So no antibodies means it's not going to attack anything. It's going to receive, in fact, AB is going to receive everything, right? So this is safe. So sometimes people call AB like universal recipient. So if they can receive blood from type O people, they can receive blood from A people, they can receive blood from B people. Okay, and O sometimes is called a universal donor. You can give it to AB, you can give it to A, you can also give it to B. So this is like a shortcut if you want. Okay, if you don't want to think about the reasoning behind it, if you just want to see, who can donate to who, you can use this transfusion chart. The arrow shows you who you can give to. Obviously, you can receive from your own kind, right? Like if you're type A, you can receive from other type A as well. Is this a little bit better, Miret? Or do you have a follow-up question?
Yes,
Okay,
Professor.
good. Okay, so let's go back to here, right? Let's take a moment to fill this in. If you are type A person, who can you donate red cells to? You can donate to other A's, and you can also donate to AB. If you are type B, you can donate to B or other AB. If you're O, then you can donate to everybody. Okay? Within the ABO blood group, right? But like in reality, you actually have to check for other compatibility issues as well, which we'll talk more about next class. What about AB? Who can you give to if you are AB? Who can you give to if you are AB?
Just A and B.
Just other ABs, that's right. Exactly. I think that's a healthy place to stop. Lots of information. Try to study for your quiz as well for next Tuesday. I'll wrap this up on Thursday. Okay. See you guys later. Have a good one. Bye-bye.
Thank you, Professor.
No problem.
Thank you.